Welcome life science learners to another lesson. Trust that you guys are well and excited about what we're going to do today. In our lesson today, we're going to focus on revising meiosis, a very important topic. It's important that you remember that meiosis is in both paper 1 and paper 2, and hence it's crucial that you understand the process well and that you're able to revise this section and work out different types of questions. In our lesson today, we're going to have an overview of the process of meiosis. We're going to try and understand the unique characteristics of each phase so that in a test or in an exam, when you present it with the diagram, and as we would work on later, you would be able to identify these stages. So, as we do in any revision lesson or when you're revising, it's important that you look at some of those important terms and concepts that are fundamental to remembering any section. And I always say that in life sciences, having a list of terms is important as you revise a section. So let's look at some important concepts as we get into the overview of the lesson, and then we will try and look at the specific processes and the different stages of meiosis. So let's get straight into it, guys. So as I mentioned, important terms. We'll review the main concepts around the overview of meiosis, and then we'll discuss some application-based questions as we get into the lesson. So, as I mentioned, terms. It's important that we draw reference to the process of mitosis, something that you've done in grade 10. Mitosis is often a process that we understand in terms of how cells produce copies of themselves. When we complete meiosis in grade 12, we often look at a comparison between the two processes. And that's again to help you understand and differentiate between what meiosis is and what mitosis is. So we've got to recognize that meiosis is a type of division that produces gametes, and it's involved in two important processes in humans. We refer to them as gametogenesis, where spermatogenesis and oogenesis produce the gametes. And so we recognize that meiosis is fundamental to producing gametes. The concept of a somatic cell and how these somatic cells are cells that have a full set of chromosomes, and the context of gametes being cells that have half the full set of chromosomes, or we refer to them as cells that are haploid. The concept of haploid and diploid again refers to cells and their chromosome number. When we talk of somatic cells, guys, we often refer to cells that are diploid or having a full set of chromosomes. Remember that full set refers to a set coming from the egg and a set coming from the sperm, and that collectively gives you a 2n complement or a full set. So refer to that being diploid. We also recognize that your gametes are cells that are haploid, having half the full set, so these would be having N and N. So we often refer to the gametes as having half the full set of chromosomes. Let's move on to some more terminology. The concept of polyploidy again refers to cells that have multiple sets of chromosomes, and we look at the advantages of polyploidy in commercial food crop production and the concept around how polyploidy happens, and we see the advantages of that to the production of food and the quality of food crop. We also look at the concept of aneuploidy, where we see there's an abnormality in the chromosome number, and often we will reference the process of how non-disjunction, or when something goes wrong during meiosis, where we have the non-separation of chromosomes resulting in a gamete that has either an extra or one less chromosome, which then eventually fertilizes to form an organism that has a chromosome abnormality in terms of the number of chromosomes. So either one less or one more would be aneuploid. The concept of being able to identify what a chromosome is, and often I use this image to use that comparison. So here we have what we refer to as a homologous pair of chromosomes. Again, chromosomes that are similar in shape, size, and have genes at the same location. So homologous. And that these contain chromatids, and so these are the structures that are produced, that are held together by the centromere, an important concept in identifying the structure of a chromosome, and that centrioles are produced during cell division, where they produce the spindle fibers. So again, we will look at those structures in the process. The concept of homologous chromosomes, as I've just mentioned, chromosomes that are similar in shape, size, and having genes at the same locus, and locus is the position of a chromosome of a gene on a chromosome. Right, the concept of karyotyping and how karyotypes are used to look at chromosomal abnormalities or the number of chromosomes, and to determine the gender of an individual. The Human Genome Project, and what does the genome of an organism mean? So it's the full set of all the genetic information in an individual cell that has been sequenced, and that often refers to the genome of an organism. The concept of a bivalent and how that forms during what we refer to a process called crossing over, when pairs of chromosomes come together and exchange genetic information between inner chromatids, and the concept of what that point of crossing over is called as chiasmata, and then finally the importance of crossing over, which we discuss later. It's important that we look at independent assortment and chromosomal mutations as important concepts in either producing variation in organisms and mutation in individuals. Cytokinesis refers to the division of the cytoplasm, and karyokinesis, the division of the nuclear material that happens during meiosis and mitosis. And then finally, the relevance of meiosis to gamete production, so the processes of spermatogenesis and oogenesis as being two important processes in understanding gamete formation. Okay, it's important that we look at an overview of the process, and we recognize that meiosis consists of two phases, meiosis 1 and meiosis 2. We often recognize that by looking at different phases in meiosis 1, and we know that there's one interphase that happens just before meiosis 1, which is then followed by prophase 1, metaphase 1, anaphase 1, and then telophase 1. It's crucial that we remember the acronym PMAT, and also that we know that in meiosis there's always a number attached to any stage. It's either prophase 1 or prophase 2, and that's again in relation to whether it's the first meiotic division and the second meiotic division. So if you write any phase without a number, and if you're indicating prophase, that again is the context of mitosis. And it's important that you recognize that prophase in meiosis is either prophase 1 or prophase 2. And so never write a phase without a number when we refer to meiosis. All right. And so we do recognize that meiosis 2, or the second part of meiosis, contains 4 phases, which are prophase 2, metaphase 2, anaphase 2, and telophase 2. And guys, as we wrap this segment up, this is an overview of the process. I'd like to spend some time getting into the overview of each process and then some questions. But before we get into that, you guys deserve a little break, stretch break, refreshing, and I'll see you in a bit.